Well, hello. Welcome back to Discipleship Squared. Today, we're going to start talking about a pretty important experiment that was done that supposedly really showed that evolution could happen by chance. So, let me share my screen here. The question we want to deal with today is, could life actually happen by chance? And what we're going to do is talk about the experiment that was done by a guy named Miller and Fox. Um, Now, remember how we are approaching our uh, videos. We're talking about origins, and there are two models for origins. There's evolution and creation. Okay, we got here by chance, or we were actually created. And remember, you can't really prove either one of those because they're historical things. They happened in the past. And so they weren't observable, they aren't repeatable in the laboratory as such. But it doesn't mean we can't use science to investigate them. What we do is we take a look at the scientific data that we see in the world today, and we try to figure out which model is supported by the most evidence. And so that's what we're attempting to do. And remember, we talked about I think the second video about the two models and how they're opposite of each other. Creation says that we were designed, but evolution says we happened by chance. Creationists say that every life was complex from the beginning. We had all kinds of complexity in just like a matter of a week when God created the heavens and the earth. But evolutionists say no, that happened from simple to complex, and it was over long periods of time. And the creationists say there were no transitional forms. Evolutionists say there are many transitional forms, and it was an old Earth, maybe millions or billions of years old, compared to what creationists would suggest. A literal interpretation of the Bible suggests maybe only 10,000 years old. So these are really opposite of each other. And what we want to do over the next few videos is really center in at first on this design versus chance. Could life actually have occurred from a non-living world by chance? So that's what we're talking about over the next few videos. It's basically biochemistry, and we're looking at the biochemistry evidence. And does it support the evolutionary model or does it support the creation model? Now, um, one of the most important experiments done during this time, it was done by Stanley Miller. Okay, he was a graduate student. He worked with a guy named Harold Urey, and he was a very studious guy, but he really was trying to show how evolution could occur. Obviously, it's kind of a problem. How do you explain how life could arise from a non-living world, just chemicals? And so he did an famous experiment. It's almost in every textbook in America, in high school biology and college textbooks and stuff. And this experiment is basically used by most evolutionists to show, yeah, life could arise from an inanimate world. So, what did the experiment do? Well, according to Miller and another guy named Harold Urey, the early Earth had a reducing atmosphere. Now, what does that mean? Well, that means it didn't have oxygen. Most people don't believe that our planet has always had an oxygen in our atmosphere. What he suggested was that it had um, an abundance in water vapor and hydrogen gas and methane and ammonia. You say, well, why would people think that? Well, if you look at the universe, there's water in some places in the universe. There's much hydrogen gas. In fact, that's the biggest amount of atoms that there are in the universe. There's more hydrogen. I think the universe has 99% of it is hydrogen. There's also pockets of methane gas and ammonia. If you look at our solar system itself, some of the outer planets and their outer moons have like water vapor and hydrogen gas in them and methane and ammonia. And so these chemicals, if you put those all together, they all have the atoms that it's needed for life. Life is based on carbon, and carbon, carbon dioxide, and we have all of the proteins that we make. They have hydrogen and they have oxygen in them. And so the idea here was, you need these chemicals probably in order to get life. And so Miller and Urey came up with an experiment to investigate that. So here's the actual experiment as it's set up. Stanley Miller made this really interesting piece of equipment here. He basically had a glass bulb here, and he put the chemicals in there that he had suggested were in the early Earth's atmosphere. So he basically had methane, he had ammonia, he had hydrogen, and he had water, and he had it all in this little part right here. And what he did was he heated it up so it would boil, so that as this came up here, this would simulate the early Earth's atmosphere. And then what he suggested was that all of these chemicals were in the atmosphere. And obviously, you could take those chemicals and shake them up all you want to in a plastic bag or in a beaker, and they're not going to make anything. They have to have some energy in order to put them together and rearrange those chemicals into things for life. And so, what he suggested, and Urey suggested this as well, is that maybe lightning, there was lots of lightning in the atmosphere. And if that lightning and these molecules happen to come together at the same time, that would provide energy to rearrange these molecules. Okay. Now, not only did he use electrodes for the lightning, and this is called the sparking chamber up here, and this was hooked up to, I don't know, like 15,000 volts, because that's what lightning is. It's a really, really, heavy duty charge. All right. And what he did was he would allow that atmosphere to be sparked with this energy. And then what would happen is it would fall down in here. And this was a cold water, what we call a condenser. Basically, they just ran cold water through this to cool off. Obviously, it boiled to get up into here, but then they wanted to cool it off. And then when it got down to here, they had what we called a cold water trap. And basically, what would happen then is if there were any molecules formed by this process of sparking them, what would happen is they would collect in the bottom, and then what they would do is they would pull those out and see what they got. So Miller did this. He ran it for, I don't know, 2 weeks or 3 weeks, something like that. And then what he did is he sampled this material down here. And what he found was that he had water in there, and he also had some very simple organic molecules. In particular, what he found was that he found a lot of amino acids. Now, amino acids are really, really pretty important in this whole process. Most of the natural amino acids that occur in nature, he actually formed. He also formed some hydroxy acids. He formed some purines and pyrimidines. If you know anything about DNA and the process of replication, those are found there. And also he made some sugars, which are also found in DNA. And so these have all been made by variants of the Miller experiment. So he mainly in his first experiment, he got amino acids. But then since that time, they've done other chemicals. They've tried to do different sets of chemicals besides just the 4 that he used. They've also used ultraviolet light in that experiment for energy. And they've also used just plain old heat. And what basically they have found is that they were able to make these precursors for living things. And for Miller's experiment especially, they were able to make amino acids. Now, amino acids are really important because that's what you make proteins out of. Proteins are just chains of amino acids. And so here, Miller was actually able to make the precursors of living things, what living things are made out of, and it could just happen by chance. Now, Sidney Fox was a guy that came along just a little bit later. Miller's experiment was in 53. In 58, Fox and a colleague of his, what they did was they said, okay, so if we got these amino acids, I wonder if we can make those somehow go together, okay, by chance to make something that resembled proteins. And so they actually did this. They took some dry heat, and they basically just heated the mixtures of dry amino acids up to 180 degrees. Kind of like what would happen is if you had these amino acids that were forming in the atmosphere fall down on, like, say, some hot volcanic rock. And what they found was that heat, that undirected energy, those chemical bonds actually formed that link the amino acids together into polymers. And polymers are chains of amino acids. And again, those are basically related to protein. They called them proteinoids. They weren't real proteins. So Sidney, Fox, and Miller didn't actually make life, but they showed that just by chance alone, and just the processes that we normally find out here on the Earth today, if those had been in place back then, and it was a reducing atmosphere, you could actually make the precursors for living things. Sidney Fox called these things coacervates, okay? And they had protein acting types of things. And so basically what happened was you have this primordial soup in the oceans being formed from lightning, and so you had these amino acids falling into that, and then you get these mixtures of amino acids, and then if they came in contact with dry heat, what Fox showed was that you could take those short chains of amino acids and they would actually form what he called protocells. And these protocells kind of acted like it was kind of like inside a membrane, and they showed some protein-like activity. Now, these are two really famous experiments, and basically what they show is that you can make the building blocks of living things occur just by chance. You didn't need a God to create them. Well, that supports the evolutionary model. And every textbook in America has this. I mean, I don't care what textbook you look, you're going to find Sidney or Stanley Miller's experiment, the picture of that sparking chamber. And you're also usually going to hear about Sidney Fox as well, and how all of this could occur by chance. And it's just assumed by evolutionists because of these two experiments and some other ones since then. But basically, it's just assumed that, yeah, you could get life happening just by chance. So it supports the evolutionary model. Or does it? A closer look at what happened in these experiments suggests that maybe this doesn't support evolution. First of all, you have this thing called chirality. Molecular models have shapes. Sometimes they're isomers of each other, they're mirror images, okay? And living cells have left-handed amino acids that form into proteins, okay? So like you can have an amino acid that's right-handed, or you can have one that's left-handed. And they're the same in terms of what they contain, but they're different in terms of their shapes. And what we find is that every living protein is always made up of just left-handed amino acids. But the experiment that Miller did, and people since then, they always produces 50% left-handed amino acids, but 50% right-handed amino acids as well. And they have never had one that just formed just left-handed proteins or just left-handed amino acids. Well, in order to get a living cell, you have to have left-handed. You can only have left-handed amino acids. So this chirality, or the shape, this isomeric property of organic molecules, is a problem for evolutionists. They have not shown at all, and this experiment, as many times as has been done, has never shown that you can make just left-handed amino acids. But let's say even if you could get the right kind of amino acids and the number that you needed, could proteins actually form by chance? Now remember, proteins are chains of amino acids. So could that actually form? Could it really form even if you did get those amino acids? Well, you also have this issue called of what we call undirected energy. In other words, you needed something to put those molecules together into amino acids. And remember, Miller used lightning. Later on, he also tried UV light, and it also worked. Some people used heat, and that worked, okay? But these are all undirected kinds of energy. They were just in the environment. And what we find out is that undirected energy, if you use it today in experiments, it's 10 times more destructive than it is productive. In other words, if you're using undirected energy and you go up a ladder one step, What happens is that energy is 10 times more destructive than productive. So every time you go up the ladder one step, you go down 9. So you never make it up the ladder, you end up going down the ladder. It doesn't get more complex, it eventually gets less complex the longer you make that molecule because that energy, that undirected energy breaks it down. You see, biochemical reactions are reversible. They approach equilibrium, they don't approach end products. And so the longer they get, the more likely they are to break back down into their original products. So the longer the chain of molecules, like say a chain of amino acids, the more likely it is to break down due to the destructive nature of undirected energy. That's just the way undirected energy works. It's way more destructive than productive. And so it's very unlikely that you could get a long chain of amino acids. In fact, most proteins, the smallest protein is like around 40 or 50 to, I think it's 52 amino acids. And most proteins are actually like 200 or 300 amino acids long. The chance of those forming just by undirected energy is basically nothing because it's not going to make these long chains. And then you have this issue about the trap. Now, most people, when they talk about this trap, and most science teachers will tell you this and stuff, they made this trap so that they could actually collect these products down here and get them down here into this, where they could sample it and they could actually test to see if products actually form. But that's not the only reason why the trap was there. Stanley Miller realized this undirected energy is 10 times more destructive than productive. And he realized that if he just kept running these things through, that there wouldn't be any appreciable buildup of molecules other than these things because they would get destroyed 10 times faster than they'd form. So this trap was actually made so that they would get trapped out of there, so that they wouldn't have this undirected energy hit them again. Now, the actual experiment then is kind of biased towards making something that might not be able to happen in nature. Now, yeah, it is true that maybe some of these chemicals could maybe, land in the ocean and then maybe float under a rock for a while and maybe grow, but eventually they're going to come back out from under that rock. Some people say, well, this happened down in the deep ocean near deep sea vents. And so they say, well, then they would be protected from, like, say, ultraviolet light and stuff. But the heat that formed them down there would be the same kind of heat that could also destroy them. And so this whole idea of the trap here is also kind of anti-productive. We don't believe that Stanley Miller's experiment, even though he formed this stuff, it actually, if you didn't have the trap, it wouldn't have formed the stuff. So Miller's experiment actually suggests that because the shapes of the amino acids that did form, they wouldn't make up living forms of proteins because they were not all left-handed. They were part left and part right, usually 50-50. And the destructive powers of that undirected energy wouldn't allow large molecules, let alone proteins, to form. So Miller's experiment actually shows that evolution couldn't occur. But it's interesting that most evolutionists, in fact, most science teachers and a lot of professors don't even know this about his experiment. They don't understand these two principles. So those are two principles that you can say, wow, you know what? That experiment, that's evidence to support the creation model. And that's pretty interesting. One of the most widely used experiments to show that evolution is true, actually, when you look at it deeper, shows that it supports the creation model, that things couldn't happen by chance. And we're going to explore that further in some of the next videos.